As we continue our discussion of the external structures of bacterial cells, besides the flagella, we have a few other ones. Just to remind you, when we're talking about external structures, we're talking about structures that actually are beyond the cell membrane. So these are going to be on the outside. Fimbriae are one of these external structures. These are going to be very sticky bristle-like projections, and they're going to be made of protein. Now by bristle-like, that means they're pretty rigid structures, and as you can see in the image here, a cell can have tons of these projecting from the outside. Now there's a few different functions that these fimbriae are going to have. One of them is that they are going to help bacterial cells to adhere or stick to substances. So this could be stick to specific surfaces. Another thing that they do is they do help with movement. The way that they do that is they are going to be involved in kind of a pulling mechanism. Think of it like an inchworm. If you're familiar with an inchworm, they kind of reach out, they stick, and then they pull the rest of their body along. So that's essentially what the fimbriae are going to do when they are involved in movement. And then another thing that they can do is that if you do have two bacteria close together, they can conduct electrical signals between those separate bacterial cells. So in that way, if they're conducting electrical signals, they would be involved in some communication between cells. Another structure that we can have is called a pilus. And this is a specialized fimbriae, so it is sticky, it's going to be pretty rigid, and this one specifically is used for DNA transfer between cells. And that DNA transfer process is what we call conjugation. So in this case, it's going to actually stick between those two cells, it's going to link them together, and then the DNA will be passed through this little connection or through this little tunnel from one cell into the next cell. So because of that, we can have some genetic variation that starts to take place because a particular cell is now getting some new DNA that it didn't have before when it does get passed through that pilus. Bacterial cells also have cell walls. So the cell wall is going to be found in most prokaryotic cells. As you can see just in this picture here, when we do have the cell walls, one thing that they're going to do is they're going to cause the cells to have different shapes to them. Because this is a pretty rigid structure on the outside. We call it a coccyx shape when it is round or spherical. We can have bacilli, and those are going to be kind of long and slender. And then you can also see we can have helical bacterial cells as well. And again, all of these shapes are maintained by that cell wall that's going to be around the outside of the cell. The cell wall does have a number of different functions that it's providing for the overall cell. And one of those is structure. Again, it's on the outside, it's rigid, it's going to help to maintain the shape of the cell. Another thing is that it's going to help protect from osmotic forces. Now, what we mean by osmotic forces is that if we have a flow of water in or we have a flow of water out, it's going to help that cell not to swell up too much so that it would burst. So it'll prevent that bursting or rupturing from happening. And then at the same time, it's going to help it not to shrink so much if you do have water that's actually leaving the cell. So we're really talking about prevention of swelling and shrinking when we say osmotic forces. And then lastly, because this is a layer on the outside of the bacterial cell, it does have some of a protective function. It is going to help that cell resist some specific drugs when we are trying to treat some that are responsible for causing some diseases. Now, if we talk more about this bacterial cell wall, it is made up of a polysaccharide that we call peptidoglycan. What you're seeing here is the peptidoglycan that's making up the cell wall. And this cell wall, it has a sugar component, which you can see the sugar chains, those are the hexagons. And then there's also these cross links that connect all those sugars together, and that's the peptido part. So it's really these two together that are going to form this cell wall. When we do see cell walls in other types of organisms beside bacteria, they do tend to be made up of different materials. So the fact that this is peptidoglycan is pretty unique to our bacterial cells. And there's really two different arrangements to the cell wall in our bacteria. So this is where gram-positive and gram-negative terms come into play. One type, okay, so type number 1 is going to be the gram-positive bacterial cell walls. And with these are going to have this really thick cell wall. So notice how thick it is right here, super thick. It's on the outside of the cell, and so this would be outside of their cellular membrane, which you can see below that. And the fact that this is so thick means that if we do perform this special type of staining technique called the gram staining, it's going to really suck up and absorb that dye. The dye's name is crystal violet. So when it sucks up and absorbs that dye, we see a violet or a purple color to our cells with if they're gram positive. This will be different from the appearance that we get when we do have the gram negative particular cells. Now, if we look at the gram negative, you can see there's quite a difference here in the overall structure of the outside of the cell. Notice that this one, that's the peptidoglycan, so it's very thin. It's a very thin layer, and it's actually not on the outermost portion of the cell. So instead, we have this thin peptidoglycan layer that's really sandwiched between two different membranes. So besides the normal membrane, cytoplasmic membrane that they have down here, we have this extra one up here on the top. That extra membrane that we have is going to provide extra protection against harsh environments. Another thing about these is that these tend to be especially dangerous or especially toxic to human beings or other animals. And one of the reasons for that is that there's some unique chemicals in this outermost membrane layer. And those chemicals are lipopolysaccharides. Lipopolysaccharides, they have a polysaccharide or sugar component, and then they also have a lipid component. And that lipid component is lipid A, and all of these that we see up here are lipid A, these little brown structures that are sticking out the top. And those do cause a number of reactions when we're talking about animals or humans. Now, if these cells are stained with gram stain, because that peptidoglycan is kind of sunk beneath, it's not going to absorb the stain the same way. So for these, they will appear pink when we do the gram stain, as opposed to the gram positive ones, which were appearing more purple with the gram staining. As I mentioned, infections by this type of bacteria are going to be especially dangerous because of that lipid A component, which again is right here, that we find on the outside of them. So extra care has to be taken with these guys, and that lipid A would be there whether the cells were really alive or whether they were killed. We're going to have the lipid A, and lipid A is responsible for, again, causing some serious reactions. We have a unique type of bacteria that actually lacks the cell wall altogether. So if they do lack this cell wall altogether, these are called mycoplasmas. Mycoplasmas, they aren't going to have the peptidoglycan there. As you can see, they've got lipoproteins on the outside, so that's what we're seeing right here. And these are also pretty tiny cells. All of our bacterial cells are really tiny. So a unique one when we're talking about the mycoplasmas. They don't really exhibit the typical characteristics that we have with bacterial cells as far as the external surfaces are concerned. All right, so that was our external features of bacterial cells. So that was everything outside of the cytoplasmic membrane. Well, if we talk about the cytoplasmic membrane itself and what its actual properties are, the cytoplasmic membrane is located beneath the glycocalyx and beneath the cell wall. This cytoplasmic membrane, the structure is what you see here in this picture. It has two components to it. It has phospholipids, and then it also has proteins. The purple molecules that you see in this picture, they're the proteins, and then the phospholipids are the ones that have the little gray circles, and then the yellow, what we call tails, associated with them. I like to think of the phospholipids as the structural part of the membrane. What I mean by that is that that's what's actually going to hold the overall shape of it together and make sure it does stay intact. But then the proteins are there, and the proteins are extremely important because the proteins are going to provide the functional part of it. So what we mean by that is they're really the ones that are going to give that particular membrane its unique characteristics. When we look at the membrane itself, the inside of the membrane, what you see here that is yellow, that part is hydrophobic. So that portion of the membrane excludes water. It doesn't want water or polar molecules to enter. And then the two exterior surfaces, where you see all the little gray circles, those are going to be hydrophilic, or in other words, they will associate with water. The overall function of the membrane, it is on the outside of the cell, And its big job, besides just holding everything in there and keeping everything together, is that it does regulate what gets to go in and what gets to go out. And we call this selective permeability. It's a little more than just semi-permeable, because when we use the word semi-permeable, we're basically saying some stuff goes in, some stuff doesn't. And that is part of it. There will be some stuff that readily crosses on its own, and then there'll be other stuff that can't get across. So if we talk about the things that can't get across on its own, so there will be certain things that cannot get across the membrane. So they would not be able to go like this. And the primary reason why they couldn't go like that is because remember that the inside, this portion right here where we have the yellow, it does not like water. Okay, so if it doesn't like water, that will exclude certain things that are similar to water from getting into that portion. Okay, so what is unable to cross the membrane? Well, the types of substances that can't get across would be large molecules. Okay, so large things are going to have a lot of trouble just squeezing right through anyway. Ions, or things that have charges to them, they're going to have trouble getting through there because they are excluded from the yellow region. And hydrophilic substances, so they would have trouble. And what we mean by these two, basically these are things that dissolve in water. So in other words, if we have substances that normally dissolve in water, they're not going to be able to get across this membrane. Now, they can't get across on their own. So there are ways to get them across, because certainly the cell is going to have times when it needs to bring some charged substances in, when it needs to bring some hydrophilic substances in. And so this is really where the proteins come into play. Proteins, one of the major functions that they have when they're embedded in that membrane is that they are going to help pull in those substances that wouldn't normally get across. And this is where they choose. So they will decide each moment of every day what they want to selectively bring in and what they want to block at that moment in time. And a lot of these proteins, they'll have like a little gate on them. So sometimes they'll open up and they'll let stuff kind of flow through, and other times they're going to block it because they're going to actually close their gate so that substances can't get through. Another thing to understand about membranes, because they are blocking the passage of a lot of stuff through, is that they are able to maintain concentration gradients. Concentration gradients are when we're able to concentrate chemicals on one side of the membrane or the other. Or another way to say it is we can have a different concentration on one side compared to the concentration that we have on the other side. So it's not going to be the same on the two sides. And we can see that in this picture here. If you just look at the little orange ones, the little orange particles, the sodium, they have positive charge. Because they have a charge to them, they're one of those substances that can't just get across the membrane on its own, because charged things can't. And so we have a lot of them on the outside of the cell in this picture, and we have very little on the inside in this picture. Now, anytime we have charged particles that we're talking about, and the green and the orange in this picture, certainly that applies to them. If we have charged particles, besides having different concentrations on the two sides of the membrane, there's also going to be a charge difference on the two sides of the membrane. So notice in this picture, we've got more positive stuff on the outside and more negative stuff on the inside. This is what we call an electrical gradient, or an electrical difference, a voltage difference would be another way to say it. And when we're talking about concentration gradients and electrical gradients, this does affect the way that molecules want to move, whether they want to go across the membrane one way or they want to go across the other direction. So if we talk about moving and selecting and things coming across that membrane, we will have two types of transport, or two types of movement to get things across the membrane. One type, the first type, is passive processes. And if you just think about that word passive, passive kind of means that you just let everything go. With passive processes, the cell does not really have to expend energy. It doesn't have to do much to make them happen. The passive processes, they do fall into different categories. And in this image, we've got 4 different categories shown here. All of them represent examples of passive processes. And the key with passive processes, besides the fact that no energy is spent, is that the concentration gradient is going to allow them to move from a high concentration to a low concentration. So they're going to go, in this picture, from, if we just look at the first one where you have a whole bunch, they want to spread out. So they're going to actually move to the region where there's not as many of them. So they go from where they are at a high concentration to where they're at a low concentration. And that's the case with all of these. If you just look across here, everything's moving from where it's really crowded towards the side. Just look at the direction of the arrow towards the side where there's not as much of them. That's passive. Now, some substances we said can just move right across the membrane, fine, on their own. They don't need any help. That's really what diffusion is going to be, when they just move right across. Sometimes we have substances that can't get across, so those hydrophilic substances, the charged substances. Facilitated diffusion is for those guys. So with facilitated diffusion, so these two are examples of that, just two different ways of looking at it. This is when the proteins in the membrane provide help. So by providing help, they're going to provide maybe a little tunnel that's going to help the substances get across. So they'll open up their tunnel, the substances flow through, and so they get to the other side. Still though, the substances are moving from a high concentration to a lower concentration, so they're just spreading out, and we don't have to use energy to make that happen. The fourth one that you see here is what we call osmosis. Osmosis is specifically going to involve water molecules, and this is going to be water molecules, again, moving from a high concentration to a low concentration. That part right there is going to be one of the kind of key take-home messages from passive processes. They don't use energy, and they do move things from a high to a low concentration. If we look closer at osmosis, we mentioned osmotic pressures causing cells to swell up or to shrink, and it's really all about what we see right here. So we have three different types of effects of solutions on cells as far as osmosis goes, or osmotic pressure goes. And so we classify these as isotonic, hypertonic, and hypotonic that you see on the bottom. So in isotonic, you have essentially the same concentration of stuff on the outside and the same concentration of stuff or solutes, which is things dissolved in the water on the outside. So it's the same in both places. So a nice even distribution of substances, which means that you don't have a rush of water into the cell and you don't have a rush of water out of the cell. Everything just kind of stays status quo, if you will. So with isotonic solutions, the concentration is the same on both sides. Everything stays the same. Notice that my cell is staying the same shape. It's not swelling up. With the middle one, the hypertonic solutions. With hypertonic, hyper means high. So what that high means is that you have a high concentration of stuff on the outside versus what you have on the inside of the cell. And the water will be drawn towards where the high concentration is at. And so water is moving out of the cell. We see that the cell starts to shrink up. If you have a cell that doesn't have a cell wall around it, this one's going to shrink even more. And you can see that it's really shriveled up in this upper picture right here. At the bottom, we have the cell that has the cell wall. You still have a shrinking of the cell membrane. In some cases, the pulling away of the cell membrane from the cell wall can be problematic. So we can see a little bit of that right here. But in general, you can have the cell shrinking, but it's not shrinking to the same extent as the ones that don't have the cell wall around them. The last solution, the hypotonic, is the opposite of the hypertonic, because with this one, the water actually wants to go into the cell because it's trying to, again, even things out. And when the water moves into the cell, we're going to see that the cell expands. So it starts to swell up. And if there's no cell wall around the outside, so no rigid box kind of holding everything together, then the cell membrane stretches, 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 and if it stretches too much, it actually bursts and it ruptures. And that is toxic for a cell if it does rupture like that. So if we're talking about a typical animal cell, or we're talking about mycoplasmas, those are things that don't have a cell wall around them, they're going to rupture. If we're talking about the typical bacterial cell that does have a cell wall around it, then they'll plump up. They plump up as much as they can to fill that rigid cell wall, and then at that point, that's just all we're going to see. They're not going to swell anymore beyond that particular point. So that's the effects of different concentrations of solute in solution. So osmotic pressure, when we're talking about things responding to osmotic pressure, That's what we're looking at. We're looking at, does everything stay the same size? Does water move out and cause it to shrink, which would be the case of the hypertonic solution? Or does water actually move in and cause it to swell up, possibly rupture, which is the hypotonic solution? The second type of transport or category of transport is going to be active processes. And with active processes, the cell does have to spend energy. That energy that it spends is typically going to be in the form of ATP. So when we look at pictures of this, a lot of times we do see the ATP being used, like you see right there. And so cell is spending energy, and then the other key thing is that things are going to move from a low concentration to a high concentration. And when we go from a low concentration to a high concentration, that's like taking something that's really spread out and trying to concentrate it or pile it really close together. Or another way to say it is it's like rolling the ball up the hill. The ball certainly doesn't want to go up the hill. It takes effort to do that. So in the same way, it's going to require energy to actually pack these things in tighter than what they were previously.